So the gist of the whole chapter, this whole chapter is about how we organize species into categorizations. You know, we talked about what a species was. You know, we talked all about that stuff. We talked about how we form species. Now we're going to talk about how we organize them in a routine, usable thing, right? When we have all these species, we've got to have some type of methodology to organize them. And that's all what we're talking about tonight. The science of organizing them is taxonomy. Okay, what makes Homo sapien, Homo erectus, Homo neanderthal? How do we group all those? Why do we group all those together? Okay, that is taxonomy. Now, taxonomy can be a matter of barroom fights in some groups. You know, what's the species in microbiology? It's bacteria. There's a debate about that. Okay, what is a species? That's a moving target. It's always been a moving target in science, at least for the last few hundred years. How do we classify something as a species? It just depends. And not only does it not just depend, we're actually not afraid to go back and change things. Because you know what? We group this with a species now. Well, we might go back and look at some DNA evidence or some other evidence. We might say, well, you know, it made sense doing it that way, but now we've been putting it in over here. Okay, so again, this is a moving target. What might be true now may not be true 10 years from now. Okay, how do we do all this? One way we do it is morphology. We look at different shapes and say, okay, this one is similar shape to this one, and we have this long laundry list of features, and we say, okay, these are similar to each other. We have now started to move toward using DNA and protein sequencing and determining evolutionary relationships in taxonomy. And again, it depends. Lots of examples of where before morphology said one thing, we put the DNA sequence through some computer algorithms, and out comes a whole new thing. We have to actually reevaluate how we did this categorization. Okay. What you got to know about this? This is a fairly standard thing. I know you have seen this in the past. It's nothing new. It is Linnaean Classification. Carolus Linnaeus. Carolus, actually, funny story. Carolus, before he became royalty, he was Swedish royalty. His name was actually Carl Linnaeus, and he was so enamored with this, you know, Latinization, he actually changed his name or signed his name with his Latinized Carolus version. Okay, and he gave us this you know, thing, and I know you've seen this before, is what? We've got domain, which is the least specific, kingdom, phylum, class, order, family, genus, and then species. You have to know that order. Okay, you have to know the order. I'm sure what's the little meme? What's that little remind that you help remember this? I know you've heard this before. What is it? King Philip. You heard this before? Okay. Came over for good spaghetti. You never heard that? Okay, well, there's other words for spaghetti. I won't say them, but King Philip came over for good spaghetti. Huh? But and I won't say the other ways to remember it. But there's some more creative ways to remember it, I should say, out there. But this is the order that we currently use. Okay, this is the least specific, which is domain. The most specific is species. Now, sometimes we'll actually have subspecies. This is actually a level a little bit more specific. Okay. Oh, and one last thing. With this naming system, when we get to talk about plants, the order is the same, but the names change a little bit. But what I'm showing you is for Animalia, everything but plants, essentially. A little bit different with plants, but just be aware of that in biology. It's a little bit different. Okay, so how do we name species? We talked about this somewhat at the start of the semester. Right, we have this binomial naming system. Everything has a two-part name. We are Homo sapien. Okay? Staphylococcus aureus. We apply this even to bacteria. We're going to be Bacillus anthracis, another bacteria. Okay, we have this two-part thing. The first is the genus, the second is the species. Genus is more specific, genus is more general, species is more specific, and that's a great little mnemonic to remember, genus being more general, species being more specific. Okay. Now we're going to talk about something called dendrograms and cladograms and clades. What we're going to talk about are diagrams that we use to classify and organize 2 or more species. Okay, because we've got 10, 20, 30 species, we have to have some visual way of organizing all these guys together. We just don't throw all these things on a piece of paper and say, there you go. We have to have some visual way of organizing. Okay, we're going to talk about something called dendrograms. You've seen these in your book already. You're going to see these a lot more next semester. So we're going to kind of cover what the whole deal is about them, how to read them, and what they're really saying. Really important, you know what a clade is. Okay, a clade is a group of species that share a set of characteristics. Okay. Really, the whole point of this, the whole thing we're trying to tell you here and show you is what? We have got 2 or more species. In many cases, we've got 10, 20, 30 different organisms, or 10, 20, 30 different species. We have to have some way of visually showing a relationship between all of them. Right? Wouldn't that be real handy if we got all these species and we could summarize the relationship between them in one handy-dandy picture? Wouldn't that be good? Well, kind of a rhetorical question, because yeah, it'd be a really good thing to do. And in fact, in biology, we do have that. Okay? So let's talk about this idea of dendrograms. And what's a dendrogram? That's what we're going to cover. Not only what a dendrogram is, really what is it saying, and how we interpret it. Okay? So without further ado, this is a dendrogram. So we got these tree-like, and we call these trees. Sometimes people just call these trees. Okay, you kind of see why we call them trees, right? So what are these things, what is it really saying? What are these really trying to show us? Okay, you're going to see this is actually from next semester's work, so when you take this thing, you're going to talk about, for example, next semester, you're going to talk about plants. You're going to talk about the evolution of plants. You're going to start with the ancestral green algae, and then you're going to see how at one point, it had an offshoot and created something called vascular plants. What did not go into the vascular plants went into another offshoot that we group in the bryophytes. And then the vascular plants formed a subset called seeded plants. Those are your roses, your flower petal plants, the ones that make seeds. And those seeded plants made gymnosperms and angiosperms. And angiosperms are the ones that make the flowers. Okay, preview of coming attractions. But what do we see? We see that there's a relationship between all these different types of plants. And these dendrograms are really good ways of showing us that relationship. So we're going to talk about each part. This is a node. See where that has that split? So it goes 1 and then 2 come off. That's a node. This is a clade. This is a clade, because remember I said what's a clade? Something with what? A clade is what? What's a clade? Exactly, thank you very much. So this is a clade because all of these are vascular plants. This is a clade because all of these are seeded plants. So that's what we're saying, is what is a clade. Okay, this is a clade because all of these are seeded plants. This is a clade because all of these are vascular plants. And that's what these dendrograms show us. Okay. And this, what I was talking about before, what is a dendrogram? That's the bottom line on it. And the thing is, there are several in the book. You're going to see this. This is not something you're going to just see tonight and then work for the test. You take this when you take the second semester of this course, you're going to see these a lot more. Okay, so you have to have an idea of how to read them. Okay, one thing with these diagrams. Oh, were you done with that one, or do I need to? Were you good with this slide? Well, no. This is just that's just me talking. Are we good with this slide? Okay. So first, we got to know about one of the things about dendrograms is they have directionality. Okay, there's a top and bottom to these guys. There's a direction, just like a sentence has a start and an end. Right? Well, these dendrograms have a start and an end. Okay, for example, this one is more of a vertical pattern, right? It starts at the bottom and progresses up. This is more of a horizontal. It starts on one side and goes left to right. Now, spins and primology, you're actually going to see we have other ways of doing this. We can actually make dendrograms in circles if we really want to. That is really handy if you've got a lot of sequences, a lot of data, okay, and if you want to, instead of having one long table that's this high and this narrow, if we can take that and make it into a circle. See what I'm saying? You take it this long, and think of it like a big piece of spaghetti, and you turn it around and make a circle. There's actually circular diagrams too. You won't see any of those in your book, but we have a lot of data. I was actually making a circular one this weekend. If you have a lot of data, you can make circular diagrams also. Okay, but the point here is what? All of these things have a start and an end, just like a story, just like a sentence. Okay. Other thing with dendrograms is it shows a pathway of successive evolutionary changes. Okay, here's the start of the sentence. Here is the first chapter of the book. Goes to the second chapter, we have some evolutionary change. Okay, that is the earliest change, the most distant evolutionary change. What happened at this point? The development of vascular plants. Past this node, or this bifurcation, we have vascular plants. We set up a new clade. Okay, time goes by, we have another change where we have seeded plants. And then time goes by, and then we have the formation of flowering plants and non-flowering plants. But what's the point? If you were to read this, you would have to say, this is the earliest change or the most distant. Let me say it this way. This happened the longest time ago. Then this happened, and then this happened. Where they are located on the dendrogram is informational and telling what order it happened at. Okay? First we had the development of vascular plants. Then we had the development of seeded plants. And then we had the development of flowering plants. So if I was to give you a diagram like this and said, interpret this diagram, I would say something like the development of this, the development of that, the development of that. You should recognize that the order of the nodes tells you something about when those things happened, at least in a relative way. Okay? Okay, so you could, that might be like a possible type test question, right? If I showed you this, I would ask you to do at least some type of basic interpretation and tell me this happened first, this happened second, and then this happened third. Are we good on that? We're clear on that? All right. Then let's talk about these nodes. You've seen these before. Remember I've been pointing those lines where we have one and then two come off? Okay, those have a special name. Those are called nodes. And that bifurcation, that split, that T-junction, whatever you want to call it, okay, that's a node. Before, you have one common ancestor, and then after it, you have two descendants. And in a lot of these dendrograms, I don't say all of them, but the ones we're showing you tonight, each of those nodes represents some major evolutionary change. And kind of think about that picture I had up before. We had one node, and past that node, we had vascular plants. We had another node, another T-junction, and we had what? We had seeded plants. We had another node, we had flowering plants, the angiosperms versus the gymnosperms. Okay, so in many of these diagrams, if we see this node or this split, it tells us, hey, something major has happened past that. Okay. And a question came up in a class, would you ever see any time where instead of 1 and 2, would you see 1 branching off into 3 or 4 or 5? It depends on the dendrogram, depends on the diagram, really. Sometimes when you're doing DNA, I do work with sometimes DNA alignments, and sometimes you'll see that. But on a lot of these dendrograms, you're going to see where it shows this broad evolutionary relationships, you would not see that. So it just depends on the situation. Okay. One last thing here, and I think we'll wrap this up on at least the dendrograms. We'll move on to something else. It's the fewer nodes that separate them, the closer those are evolutionarily speaking. The more nodes that separate two groups, the further they are from each other, evolutionarily speaking. And I have an example of this, and then you'll see the point. But really, that kind of makes sense, though, doesn't it? If every node represents a major evolutionary change, then the more evolutionary changes between two groups, the more distant they are in relationship. The fewer evolutionary changes between them, the closer they are evolutionarily. Okay, so for example, people still write here. For example, right, I want to give you an example of this. I know you're writing, I'll give you about 20 more seconds. But I want to show you an example of this. I'm not going to say this was the example you're going to see on the test, but this would be a type of way I'd ask it. Okay, and it really just is an application of something you have to know. So, we have this diagram, right? Again, this is one of those vertical dendrograms. Same thing, we're talking about plants, right? We have the vascular plants here, we have the seeded plants here, and then the angiosperms are the flowering seeded plants. Which is more related here? Okay, which is more related? Well, number 3 and number 4, they're more related. Why? Because is there more nodes separating them or fewer nodes? Fewer. One node separates the angiosperms and the gymnosperms. That tells us that they are more related than are the bryophytes to the angiosperms. Because how many nodes separate angiosperms and bryophytes? Well, we count all these. Let's just say that 1, 2, 3. The more nodes separate them, the more distant their evolutionary path is. Okay, where these might have come from a common ancestor 10 million years ago, these might have come from a common ancestor 150 million years ago. And I'm just putting those numbers in there just to give you a comparison. Okay. Nothing here says that those are the numbers. I'm just pulling those numbers out of the air. But the relative size of those numbers is comparable. See, one's 10 million, one's 150 million. Okay. And that's it for the dendrograms. Now let's talk about comparing body parts. New topic, new discussion here. We now want to look at comparing body parts, wings, arms, bones. Because this tells us also something about an evolutionary relationship. Okay, so the first thing we're going to talk about is morphological divergence. And again, what's the major point here? What's the take home here? And the point is this: as scientists, how do we look at, before we started all this, we said one way we classify organisms is based on their morphology. And this dovetails with that. How do we look at morphology of something and categorize it? What do I mean by that? We look at a cat, a dog, a mouse, and a human, and we do some anatomy on them, and we look at the different bone structures, and we say which is more related to which. I measure the length of this these phalanges in a dog. I measure the phalanges in a human. I measure the size of these in a cat. I then can use that information and make a determination and say, well, a dog and a cat are more evolutionary related than a dog is to a human. Okay, that's the whole idea of using morphology. We're looking at the shapes of structures. That means bones, muscles in some cases. We're looking at how these are shaped, and that tells us something about how they're related. Okay, morphological divergence is a body part that comes from a common ancestor. But over time, that body part has become specialized to that organism. For example, here's a cat, human, whale, and bat arm. Are there common structures to those? You bet. Okay? We all have phalanges. Okay? We all have metacarpals. We have carpals. We all have the same bones. But, you see, what's the difference? Is there a difference? What do you think? You tell me, is there a difference between these bones? Are the phalanges in a human different than the phalanges in a whale? You tell me. Well, they are, obviously. So what is a scientist, let me ask you this. Which do you think is more related? Looking at the morphology here, the blue are phalanges in all of these. Which are more related? Humans and cats or humans and whales? What do you think? Maybe humans and cats. That makes more sense to me. All right? These all have common structures. But just looking at it, heck, it just looks like humans and cats were distantly related, but we're probably closer in relationship than we are to whales. Okay, that's the idea of morphological divergence. And if we could somehow have a DeLorean in a stainless steel and go back a couple million years, we would see that there might be an organism, an animal that is our common ancestor that had these features. And there is one of those nodes, one of those splits. And over time, we developed these, the cat developed these. That is morphological divergence. We start with a common ancestor, and then as time goes on, evolution happens. different species form, different evolutionary pressures on those species are applied, and then we just get different structures that are similar. Okay, the opposite point of this coin, we flipped the coin, we talked about the head side, now let's talk about the tail side, is morphological convergence. Okay, just because we see two structures that look similar does not mean they develop from the same common ancestor. A great example of this is we think about the wings of a bat and the wings of an insect. Okay. What led to the development of wings to an insect and what led to the wings in a bat were totally different processes. We would call those analogous structures. Okay, so the wings on an insect, the wings on a bat, they do the same thing, right? They're going to cause negative pressure underneath, and they're ultimately going to cause flight. Well, they do the same thing, but do they come from a common ancestor? And the answer to that is no. At least the wings did not develop from a common ancestor. So those are analogous structures. They are performing the same duty, but they come from different evolutionary lineages. But that, sometimes an idea is just so good that multiple people think of it, right? Well, same thing with evolution. Sometimes an idea is just so good that it is come up with independently of one another. Right? Think about like the wheel. Having a wheel, two wheels and moving something. That seems like a pretty good idea. Did one person think of that and then spread that knowledge? Well, probably not. Probably lots of different groups thought, oh, that's circular. Yeah, we can make a wheel. Right? Doesn't mean those are related. Well, the same thing here. Some things like wings are such good ideas in an evolutionary context that they will develop independent of one another. Okay, we're good with that? So you should know what convergence and divergence are, and you should have an idea of what analogous structures are. Okay. Talk about embryos. If we looked at embryos, human embryos, chick embryos, cat embryos, you name it, you're actually going to notice in early development, we look a lot alike. Believe it or not, all of us at one point had a tail. Now that tail went away over time, but what happens, and the idea is this, early on, before we have a lot of differentiation, remember we talked about genes turning on, genes turning off, and causing differentiation. You kind of remember this? Vaguely? And we said that what makes a skin cell, what makes a hair cell, is that some genes are turned on, and a lot of genes are turned off. Okay? But before all that happens, when we're in this embryonic stage, we look very similar to other embryos before development happens, before specialization happens. Now, here's the interesting thing. You know that before you're born, you had like webs between your hands, right? Did you know that? Web fingers? Vaguely even heard of this? Okay. The reason you don't have web fingers actually is a process called apoptosis. The cells that make up the skin between them, they're programmed to die. Okay. In development, for us at least, those cells essentially kill themselves off. They release lysozymes, they lyse themselves, they break apart inside, and then we get our little separate fingers. In some cases, that doesn't happen and people are born literally with webbed fingers. But the point here is what? That development, differentiation makes us what we are later on. But at least in early development, we look very similar to most every other embryo out there. Okay. Differential growth patterns. The idea is this. If you looked at a chimpanzee infant skull and a human infant skull. You know, are they going to look similar? I don't know if you ever had the pleasure to look at them, but if you looked at them, they have a very similar appearance. Hard to tell them apart. Okay, but what happens is that over time, a chimpanzee is going to go through different developments, and their jaw is going to come more out, and their skull will develop in a different pattern than a human skull will. And we see this a lot of times with closely related species, that over time, right, when you're at an early age, there might be very little morphological difference. But as time goes on, as time moves on, development happens, and there we see some more pronounced morphological changes between the two. Okay. And then the last part here for this chapter, and then we will wrap this up, is comparing DNA and protein. Now, when we started this, I said that what? One way that we have always categorized species is we will say, okay, these have these similar characteristics. These plants have leaves that are pointy. These leaves have pubescence on them, like a fuzzy fur on the bottom, and these don't. These are evergreens, these are not. That was how we classified them a lot of times before, and we still use that methodology. But in the last 20 or 30 years, we've actually brought a new thing online where we look at the protein and the DNA sequence of these organisms, and we try to make inferences based on that. Okay? And the idea of this is pretty straightforward. If I take sequences and I see a large difference between similar genes, the sequence that makes them up, I can say those are more distantly related. If I see fewer differences between those genetic codes of those two organisms, then I say they're more closely related. Okay. Now, how do we do that? We have a process we call a DNA alignment, or in some cases a protein alignment, where we put these sequences in a computer algorithm, a program, and the program will grind through the data, and they will put them in an order where it's informational. Okay, but what's the point? What do I want you to know? We look at the DNA sequences of organisms. If two sequences are very similar, they're more closely related. If two sequences are more dissimilar, they're more distantly related. And using these methods has really caused I would define it as a revolution, at least in taxonomy, in the last 20 years. It has caused a major change. In some cases, this has been the foundation of moving one organism from an entire different kingdom to another kingdom. And we have all these reorderings where the molecular data somehow says something totally different than the morphological data. And which is right, it depends, it really does depend. Some people want to go with more morphology, some people want to go with this more genetic difference and look at those. Which is right, it just depends. It just depends. Well, bless you. Okay, so I'm going to go forward for 1 second here and just show you what I mean by these alignments. Okay. This is a DNA alignment. Okay. All of these, you're familiar with the A, T, G's, and C's, right? So what we did was we put all these sequences in a computer program, and we let the computer move these things around, and we see that what? Do you see how these two organisms have what? A T here, and this organism here also has a T. Do you see that? But these others don't have a C. You guys see that? Okay. When we have a phylogenetic tree, we have a tree, that dendrogram, which ones do you think are going to be closer? Do you think these guys with the T are going to be closer on the tree? Yeah, they will be. So let's look at the tree we make with these. This is the tree we made. These two had that T, At the top is number 1 and number 2, goes here and here, and then the 5th one is right here. You see how we grouped those more closely because they had a commonality that the other ones did not. That's the point of these molecular techniques. That's the point. That the thing at the top, we call that an alignment. And how do we make these trees? We have algorithms, we have computer programs that we actually physically put the stuff into. I've done this a few days ago. We put the sequence into it, and we let the computer tell us the outcome. It can take a DNA alignment with I did a DNA alignment with 800 sequences, and it took about an hour and a half. It's a computation, and this is on a fairly good quad-core computer. I mean, it's a fairly good computer, and it took a couple hours to do. But it's a fairly computationally heavy process. Okay, so let me just finish these up, and then we will be done with it, and I'll give you a little word, a proviso here. Okay, what do we use? What can we use as a basis for doing this? One, we can use protein sequence. Okay, we can look at the amino acid sequence of common proteins as a way of doing this alignment. We can also look at DNA sequence. Do you think the DNA and the protein are going to be the same? Are those going to give us the same data? What do you think? Let's see this. Remember we talked about the wobble theory? What was that all about? Anyone remember that? The last test? Remember we looked at that codon table? Vaguely remember this? You had that big table at the end of your test, and you had to pick, codons and say this ATG does this protein or amino acid. Remember that? Well, let me ask you this. Do you remember how you could have multiple codons coding for one amino acid? Yeah? Point is this. I can have different protein or different sequences, DNA sequences, code for the same amino acid sequence. So if you do amino acid sequence, it's going to hide some of the genetic differences because more than one base can code for a protein. So point here is what? We have two macromolecules we can use to do these alignments. One is protein sequence and the other is DNA sequence. They both have drawbacks and they both have benefits. It just depends on what you're doing. Now, that wraps it up for tonight, but let me show you one little last point with these because we talk about them being this foolproof, absolute, lead pipe cinch thing. But they're really not. You have to be able to pull back and analyze the data. Okay, so we're doing one of the DNA alignments. Right here is Bibb Graves Hall. That's Bibb Graves Hall, right? Someone here, she was working with something called tardigrades. You ever heard of water bears? You ever heard of water bears? Very little, bless you. They look like little bugs. And don't push your pens away. We got a quick little quiz here after this, open notes. They look like little bugs, right? We got this little bug here. You have to use a microscope to see it. Okay, you can collect these from moss. Okay, so Bibb Graves. When you walk out, there's that tall stone wall next to Bibb Graves? Vaguely? You walk out the door and there's that really tall, it's about 15 feet stone wall. Okay. There's that stone wall. Girls here are from Taiwan. Here's Taipei. Oh, I spelled something like that. Taipei, Taiwan. And she goes to her father's university. He's a professor in that country. And she collects moss and collects tardigrades. So, Here was the thought. Let me draw. These are the Taiwan tardigrades. And school spirit here. Here's the JSU tardigrades. And they had some sequence from France also in here we got from NCBI. And we got these little tardigrades, these little bugs. Okay, some from Bib Graves, some sequences from France, and then some that she got from Taiwan. And you would think, what would you think? If I looked at these, at the start, the assumption was what? These were closely related, and these were probably closer related, and these were closer related, but if you looked at all of them in a DNA alignment, what would happen? These would group separately, these would group separately, and these would group separately. Does that make sense? Well, the thing is, when all the data was analyzed, what did we find? We found that this one from Taiwan was more closely related to the one from Bib Graves than it was to one from 20 meters away in Taiwan. Yeah, that was a good one. So anyhow, but you see the thing is sometimes these molecular things, they don't answer a question, or you get some of these confusing answers. So I don't want to imply that, molecular is the way to go. Morphological still has a point. Sometimes, you have to choose something that has enough difference to make a bit of difference. The problem really was there's so few differences between these, one base or two bases out of hundreds. We call it conserved. It was really not informational. Okay, so I just to say, we talk about these things, but we never say, oh, here is an error. And you're not responsible for knowing the specific error, but guess what? It ain't perfect. It simply is not perfect. Sometimes it gives us crazy data. And something that's literally, if you look on a map on the globe, this is literally from the most distant location from this location. I mean, here's the globe. We're here. These are from down here. You can't get more distant than this. Literally the other side of the planet. So, it ain't perfect. Usually it's pretty good, but it is not 100%. Okay, so the question is,